Okay, welcome back. My name is Jesse. I make GAMSAT videos, as you probably already know, unless you're new to the channel. Today, what we're going to be doing is going through the second of the Biology Crash Course series. I'm going to try and smash this out relatively quickly as we get towards the end of the year. I'd like to have all the content stuff covered before the Christmas period. I am actually planning on taking a short break from this as well, as you probably expect. It's a really long year. I think I've lost track now, but my last day off was January 15th. So it's been a really long year. And so I'm looking forward to taking just a couple of weeks off at the end of December until very early January, and jumping back into it. I'll probably still be making some videos, but probably not as many as I am at the moment. And so I'd like to get all the content out of the way so that if you are planning on prepping over that period, then you've got everything ready to go. So what we're gonna do in this one is cell structure, the plasma membrane as well, because I noticed that comes up a lot in GAMSAT. The other thing we're going to go through is the cell cycle and some quick things on cell division, but not the full theory, because I don't necessarily think it's 100% relevant. You just need to know some of the key takeaways from it. So we'll move through that pretty quickly. And then as well, we're going to go through a little bit on DNA structure. Don't want it to be too laborious a video. I've got some questions at the end as well that are ACER style. That's the big difference with this crash course compared to the physics and the chemistry ones. Trying to make sure that you're taking the biology content with a GAMSAT context in mind and looking at how it's actually applied. You don't need to know absolutely every finite, like every tiny little minuscule detail of biology, because it's just a gigantic topic. They really just want you to take away some key principles that you can apply and use to make assumptions and reason with the questions. For the most part though, they actually give you the information that you need in the STEM. The other thing is as well, it's been pointed out to me that it could be a little bit confusing navigating all of the material that's on the channel at this point, because there is a lot. So if you're from a non-science background, that's probably why you're watching this right now, or you don't have a biology background. So it's a good place to start here with the Crash Course series, and I've created playlists for each of the three. I may end up also doing like a Crash Course one for maths as well, because that seems to be a pretty big one, and a lot of people have a lot of questions about how logs work and blah blah. So starting at Crash Course, these are probably the entry level difficulty stuff. I'm also going to be working on an intermediate level that's going to bridge the gap between this and the Section 3 walkthrough videos. So you would go Crash Course first, you would then kind of like graduate onto the intermediate level one, which is kind of question analysis and a lot more narration from me. And then the final stage is the sample walkthrough questions. Those are pretty much at GAMSAT level all the time. And I usually make mine a little bit tougher than the general mix of ACER stuff. ACER have a mix of kind of pretty straightforward, simple ones, and then some more complex ones that require a bit of deeper thinking or a little bit more speed work in it. Most of mine are kind of pushing towards that difficult end to really challenge people in the way that they apply their strategies. So if you've kind of been doing both and you've been finding it difficult, that's fair enough. There is quite a big gap between the two. So I'm working on bridging that, and I'll also be posting as well on the community page, if I haven't already, a summary of what I've just said, basically. Just navigating so that you can know how to kind of navigate the content on the channel. Anyway, so we'll dive right into it. We've got a lot to get through. So the first thing that we're going to do is look at cell structure. And so in order to start there, we have to think about what the cell is actually made from. So normally we draw them as a blob, but obviously they can be any shape. It doesn't really matter. And this border here is going to be our cell membrane. Feel free to pause at any point in the video if I'm moving quite quickly, just so you can make notes and stuff like that. But I'm going to move at a relatively rapid pace to get through all the content that we've got to get through. So the cell membrane, often referred to as the phospholipid bilayer. And we don't need to really know a huge amount about the content here, but more so just understanding what this means. So when we actually kind of zoom in on this, let's do it like this. What we actually see is a double layered structure. And the idea is that it's a wall. It's got all these little circles like this facing on the outside and on the inside as well. So on either end. And what these are phosphate groups here. If you wanted to go into the chemistry of it, it's a phosphate with 4 oxygens attached like this, although they can be double bonded and things. And these are actually very charged, so they're polar. We've talked a little bit about polarity before. So therefore it is water loving. And so they'll direct themselves towards where the water is, which is going to be inside the cell and outside the cell, because most of what constitutes that fluid is water. And then hanging off them, you're going to have these little lines, tails. Again, these are actually fatty acids, and we've seen those in some of the chemistry walkthroughs and things. They're a long chain of carbons with a carboxylic end, and so often like this. But because they've got so much carbon on them, they're generally non-polar, therefore they prefer other lipids, or they prefer, they hate water, they'll try to avoid it, which is explaining why they then direct themselves inwards away from the water and protect themselves like this. And then the other side has exactly the same thing as well, right? And I could sit here and draw every single one. There's supposed to be 2 on every one, but I'm just going to cheat a little bit and do that. Hopefully I've gotten it roughly right. So we've got this kind of hydrophobic region, meaning water hating, and it tries to expel water from that region there. That is our membrane, that's the thickness of the membrane. But then we also have as well, I guess the way that I've drawn it, this is inside and this is outside, and there's just a bunch of water in that fluid, and that's why those polar phosphate groups are facing that direction. If you put the name together, phospholipid bilayer, so phospho at the top, lipids there, and then, well, it's got the lipid structure, and it's a bilayer, it's layered in 2 layers like that. All membranes are made of this basic structure. What we really care about in that is that the membrane is semi-permeable, meaning that it can choose or can it control what gets across and what does not. So you'll notice that it's pretty tightly packed in terms of the actual compounds. So what that means is that only really small things can kind of squeeze through the gaps. So if we were to kind of zoom in here, there'd be tiny, tiny, minuscule little gaps like this that it can fit through, and you could transport things across the membrane so long as it's small enough to fit through. The other thing that it has to be is it has to be non-polar, because it has to be able to be withstood while it's moving through this region. So things that are polar are going to have a bit of a more difficult time getting across, because these fatty acids are going to effectively, like a magnet, kick them back out like that. So generally speaking, if we're trying to transport things across the membrane, then we want them to be small and non-polar. Obviously, you don't always get exactly that combo. You'll notice that water, you'll see that being talked about as being moving across the membrane pretty commonly, and water is actually a polar molecule. It's just that it's so small that it kind of goes, like, we'll say undetected. It's small enough that it can bypass it still. If you had something that's large, even if it's non-polar, it may be so large that it's still being kicked out. So even though it's not perfectly non-polar, if we looked at something like glucose, for example, it's a gigantic molecule, it's got many, many carbons on it, and everything else hanging off of it, it's too big to fit through, so it requires some other help, which is then what gets us to the next bit, which is membrane transport mechanisms. So when we're trying to transport things in and out of the cell, we have a few different options here. And so I'll just try to see if I can cheat this a little bit. I'll just do this again. There's supposed to be 2 for every single circle. And let's see if I can. There we go. Cool. I'll just do that. There's our bilayer now. So the first thing is that if something can move straight across the membrane, and I might actually copy this to make it a little bit bigger, like this, then it might be able to just transfer itself right between the gaps, fit through because it's small enough and it's non-polar, or it's polar but so small that it can fit across. This will be things like water, this will be things like sodium, potassium, small ions and that kind of stuff, some small gases, oxygen and so on, can get across. And so what this is referred to as is diffusion. So whenever you hear that word in any of the questions, it just means something just passively moving across the membrane. The trick to it is that it'll diffuse from where there is a high concentration to where there is a low concentration of that thing, right? So we've got to be really careful, because I've actually put water here and I might remove that. Water gets a special name. Water follows osmosis, which is just diffusion of water. But since we're talking about high to low, water moves from where there is high water concentration, which actually means low concentration of everything else, because it's so much water that it's diluting it. So water would actually move in the opposite direction. If there's low amounts of these salts and gases and things, then water will move where there is lots of water, so high water to low water. But they always go high to low no matter what you're talking about, and that would be osmosis in that case there. So the idea is that it's following what's called a concentration gradient, and a concentration gradient is the relative difference in concentrations of any item on one side of the membrane to the other. If it's got a value of 10 on one side and a value of 3 on the other, it's going to go from the 10 side to the 3 side until they're roughly equal in concentration. It'll try to equal out, and then the rates of the transfer will be roughly equal, and it'll stay pretty steady and constant. That's the concept of diffusion. None of this costs any energy or anything. This will happen normally. The second type that we see, though, if it's too large or it's polar or for whatever reason it can't do one of these things, then what it will use is it'll use facilitated, and we don't really need to know a lot of these technical terms, but more so just understand that what actually will happen is it'll use a protein that often embeds itself inside of the membrane, like this, and the protein's job is to allow things to transfer through. So it effectively, like, it might open up a tunnel or something to create a big enough gap so that the large molecule can get through, or it might create a polar region that makes that item happy to transfer through. And again, it'll go along its concentration gradient from high to low still. That could either be into the cell or out of the cell. Then finally, what we have is active transport, and active transport is where you're actually forcing any item, whether it's small, large, polar, non-polar, against its concentration gradient, pushing it in the direction it does not want to go. That is from low concentration to high. So I said here there was low sodium on the bottom side here. So if there's low sodium and I'm trying to push it this way, then I'm going to have to pay energy to do that if there's high sodium over here, right? Because it's trying to balance out, and what I'm doing is pushing it further away from that. And so that in the process, you'll have to spend energy, which comes in the form of a molecule called ATP. You'll see that a few times, and we'll talk a bit about that a little bit more in a metabolism topic. But for now, think of that as your currency of the cell. It's the equivalent of the dollar. It's a measure of energy, and it'll expend that energy to help force the item to go in the wrong direction uphill. Really simple example is just think about a hill. If you've got a ball on the hill, it'll roll down. So naturally, without any energy added, it'll go from high to low. That's our standard diffusion and transport like this. If you're trying to push it up the hill, you have to put energy in to push it up the hill. So to go from low to high, you have to spend energy. This is referred to as active transport. And there's usually proteins involved with that, but we don't need to know anything about them. They will explain it in the STEM if it's relevant. Cool, so that's our transport now. So the membrane is really just housing everything, and it's allowing things to get across, get in, get out, based on its characteristics, or based on some protein that's acting as the gatekeeper in some way. The next thing really quickly is just to be able to identify some of the organelles, which are the little kind of organs of the cell. And we don't need to go through all of them. There's only a few that would be worth recognizing. So the first one is going to be the nucleus, most obviously. And this here contains all the genetic information, right? Contains all the DNA. And so that's obviously a pretty important one. You'll often find it just like anywhere. It doesn't have to be in the middle. So if this is our cell, you might find the nucleus like sitting over here. Often it's drawn as a kind of darkly staining region, but again, I don't think they would expect you to be able to keep up with a particular type of notation. The second thing that we have is the ribosomes, and we see these talked about a lot. They're often represented as dots like this, and they're just kind of everywhere. Their function is to make protein. That is their whole function. Then after that, we have the endoplasmic reticulum, which is a little bit of a ridiculous name, bit of a tongue twister, often referred to as ER. And this is the manufacturing plant. So think of this as like the factory, the manufacturing line of the cell. So it makes all kinds of stuff. It makes carbohydrates or sugars. It makes proteins. It makes lipids. So it's where all of the manufacturing occurs. And then you've got, always right next to it, you've got the Golgi apparatus. And so this is, its job is P&P, processing and packaging what comes out of the ER. And so if I gave it an analogy, it would be kind of like if you had a conveyor belt like this, the assembly line, this is the endoplasmic reticulum, putting things together, right? So you got a piece here glued to this piece, and then now you've got like this, and you get a little block, and it's moving along, and you're making some product. It dumps off the other end, and then here you've got a box ready to catch it. Here's one, maybe another one went in, and then we're going to close the lid, box it up, and ship it off like that. And so this is what the Golgi apparatus is really doing, right? You can think of it as like the Australia Post of the Australia Post of the cell kind of thing. There we go. And that might be to send it out of the cell, or might be to move it to another part of the cell. In terms of what they look like, endoplasmic reticulum, you'll normally see it as like a squiggle. I usually draw it as kind of like a scroll of paper. You can draw like vertical lines like this. There we go. You might see little dots on it. They're just ribosomes. Some parts of it have ribosomes on its surface because it's making protein right there on the surface. And the Golgi apparatus is usually drawn as like distinct little bundles like this, little blobs, so actually separate. And the idea is that things are being transported through like that and then out of the cell. That's just in case you need to kind of identify them in a cell diagram. It's pretty unlikely though. We just need to understand their main functions. And then finally, if we're looking at a plant cell, then we need to know that they also have chloroplasts. And so chloroplasts, again, I'll just draw them in just in case. So sometimes they're drawn like, almost like little chicken scratches kind of thing like this inside. Other times they look like they've got little like floorboards like that in them. But the idea is that we just need to know these are responsible for photosynthesis. So they can harvest the energy from UV light and then store it in the form of ATP and other molecules and that kind of thing. So energy storage, or energy. We kind of say in biology energy production, but if you go to chemistry and physics, the 0th law of thermodynamics says that you can't produce energy, but transducing it, right? Changing its form from sunlight energy into chemical energy. But that's obviously plants only. And then I guess one more thing that would be important to know is in comparing animal versus plant cells. Animal cells have, which is including human cells, we have a cell membrane, but that's it, which means we don't really have a whole lot of structure to our cell shape. Whereas with plants, they have a cell membrane and they have a cell wall. And the wall is a rigid structure made out of mostly carbohydrates, and it's usually drawn like a little rectangle, kind of with branches like this, and they kind of tessellate together like brickwork, and the membrane is kind of attached to the inside of it like this, and it kind of holds it out like a tent, and gives it some really rigid structure. So generally you can pick out plant cells because they look like they're tessellating like this on pattern. Animal cells just kind of float around like a big mess, like a fried egg kind of thing. Cool. Then we've got the cell cycle. So looking at what's actually happening in the life of a cell, and this includes cell division. So if we draw out, often the way that it's represented is a series of steps. So we have G1, we have S, it's a bit messy, but oh well. We have G2, and then we have M phase. And what these all stand for here, G1 is we have G's are checkpoints, which is where the cell is preparing to do things. So in this case, it's preparing for DNA replication, and it's also maintaining its size and that kind of thing, and repairing damage and that kind of thing, so maintenance. Then we've got synthesis here for S, and this is where you get DNA replication. And so we should see that the amount of DNA quite literally doubles in size at that point. And then G2, this is where now it has a checkpoint to check that synthesis went correctly. And then what it does is it prepares for division. And that includes preparing its like cell membrane to get longer, because it's going to have to elongate and then snap into two cells and have enough for it. And it's preparing for the doubling of all of its organelles and everything else as well. And then M is the true division, and depending on which type of cell, it's either going to be mitosis or meiosis, which we're going to very quickly compare. And that's where the M comes in. Now we kind of zoom in on what's happening here, and let's start with mitosis. This occurs in non-germ cells or non-gametic cells. I should say germ cells, really. Non-germ cells. You don't really need to even know what that means. It just means not the cells that are producing either sperm or egg in humans. So skin cells, heart cells, liver cells, pancreatic cells, all those are non-germ cells. They all undergo mitosis, and it's kind of like a cloning event. So what we have is, you can remember IPMAT like this. You don't need to memorize the whole process. It's really just so that you're familiar with all the words. I stands for interphase, which on the cell cycle is this region here. It's everything outside of the division. Then you've got prophase, then metaphase, anaphase, and telophase. And an extra step at the end of all that is cytokinesis, which is where the cell actually divides fully and separates its membrane. So the idea is that in interphase, I'm going to draw one chromosome representing all of our 46. So normally we have pairs of chromosomes. We have 23 pairs. In fact, I might draw a chromosome pair here. So in interphase we have, here they are, here's pair number 1. So just imagine times the number of genetic information in that by 23, and bang, you've got a full nucleus. So what's going to happen is, during interphase we have synthesis, and so this is where we'll see the DNA get doubled and you'll get these X-shaped chromosomes. The important thing is that the number of chromosomes actually doesn't double. We just change what a chromosome actually is. So we were first referring to this as a chromosome. When we double it, we're literally just like carbon copying the two arms of it into an X-shaped chromosome, and we're calling this a chromosome now. So we should still have all 46 of them. And instead we call one of those halves a chromatid instead, right? Again, you wouldn't need to know that word or anything. They would probably explain it. It's just for familiarity. So the more important thing is the numbers. The number of chromosomes actually doesn't change. The amount of genetic information in the cell does, it doubles, but the number of chromosomes does not actually double, because we redefine what a chromosome actually is. Then we go through a series of steps, and I'm not going to go through all the technical bits and pieces because the chances of you needing to know that is so slim. More importantly, what we want to know is, in mitosis, the chromosomes don't line up in pairs, they line up in single file like this, which roughly happens at metaphase, and then they will get ripped apart and pulled to either side. That is what's happening in anaphase. So they kind of get ripped away like this on these little strings, and like this. And then finally in telophase, we're starting to prepare to separate the nucleus, and then in cytokinesis separate the cells. And so we end up with 2 separate nuclei, and we will have the exact same information in either of them, assuming there were no mutations, and we end up with something that was just like the parent cell as well, ignoring the pink replicated bit, like that. So we've effectively doubled it, right? So we get 2 cells, we've doubled the cell number at that point. And so what that means is the amount of cells, the number of cells, can be represented as, if we want to get into some maths, 2 to the power of the number of divisions. So if they said something like there were 8 divisions, like that, and they said how many cells would there be, it'd be 2 to the power of 8, right? Let's just do a bit of maths here. This would be like 2 to the power of 4 squared, let's say, which is 16 squared, which is 16 times 16, which you could do as 10 plus 6 times 10 plus 6. You could even use like perfect square patterns if you wanted to do that. So it's 100 plus 60 plus 60 plus 36, which gives us 220, 256. And there we go. You could also just kind of work your way through 2 times 2 times 2 times 2 and get to the answer. There'd be 256 cells after 8 divisions and so on. So that's a really good mathematical relationship to have up your sleeve as well. But more importantly, with mitosis, what do we get out of it? First of all, we got 2 daughter cells. They were genetically identical, assuming that there were no mutations. And notice as well, the parent cell does no longer exist. The parent cell gives itself to the 2 daughter cells in that case, right? These are really the key things that we get out of mitosis. If we compare that then to meiosis, I might just do this. In meiosis, this is in germ cells. So this is in cells that are producing sperm or egg. And what actually happens is we want half the genetic information. So germ cells. So we have our parent cell, and again, I'm going to do a human cell, but just one of the chromosome pairs. So here it is, like this, and I'm also going to color code this as well, actually. So obviously we have pairs because one is from the mother, one is from the father. So I'm going to do red, that will be from the mother, and then we'll do purple, that will be from the father, like that. And then we're going to see what actually happens and trace it all through. So it goes through 2 stages of division. The first thing it does is it replicates its DNA. So let's go ahead and animate that in. It's going to replicate, bang, bang, and it's also going to replicate the other chromosome, and it becomes X-shaped. There we go. So we've still got 2 chromosomes. They're just X-shaped chromosomes or replicated chromosomes. The difference is now when they line up, they actually line up in pairs rather than single file. And so what that means is if we zoom in again on what is now happening here, I'm going to draw them really big. They cross over slightly. They link arms like this, and they swap a little bit of DNA like that between them. And so you get these recombined chromosomes where you'll have, do this to the best of my abilities here. A little bit of the purple goes to the other one. This one gets a little bit of the red. This one's mostly red, like that. And then what's going to happen is they're going to be separated. So now what's going to happen is they're going to be separated into 2 cells like this. We're going to now rip apart the partners like that, but they're already recombined. Then what we're going to do is divide them again here and here. And you can see that the genetic information in each of them is going to be slightly different. You'll see that in the first one on the left there, it's all red. The second one is mostly red with a little bit of purple. The third one has got a little bit of red. So they're all genetically different from one another, but they all have 1 chromosome, like that. So 1 chromosome, but all 4 are genetically diverse. And if you compare that then to what we had in the parent cell, it's also half the genetic information. Before we had a pair of chromosomes, now we have just 1, and it's different to the parents, and they're each different to each other. So what that means is that we can summarize. So now we get 4 daughter cells, and we get genetically different. That's all we really need to know at that point, and then understand that this process here is recombination, which is a big factor in why there is so much genetic diversity in sexually reproducing organisms, including humans. The other thing is we can get that mathematical relationship. The number of cells would be 4 to the power of n, like that, right? Ignoring anything like egg selection and stuff like that in females, we'll just assume 4 to the power of n, and you could apply any maths to that as well. That's the main thing. Now, the reason why we want half the genetic information is because then if, say, this here were a sperm cell, and then we had an egg cell, and the sperm was going to fertilize the egg, they're going to actually join the genetic information. So you want to build up the pairs rather than have 2 pairs gluing together, else then you'll have 4, and that's not actually sustainable or viable. So that's why we halve the genetic information. And I guess we can put that in as well. It's half the genetic info. There we go. Cool. So then the next thing is DNA structure. And again, this kind of bridges a little bit with some chemistry here, so I'll give as much detail as possible. The first thing we're going to look at it is we know that it's a double helix, which often is drawn like this, the ladder structure. And we've got our sugar phosphate backbone, which we'll get into more detail on. And here we've got the hydrogen bonds between the bases. And now let's actually look a little bit more at what's actually happening. It's made up of something called nucleotides, and a nucleotide is often referred to as the building block of DNA. And they're made up of 3 parts. So they're made up of a sugar, which is normally a pentagonal kind of shape, a phosphate group, which, well, I'm just going to do this for now, and then a base. I'll just draw that. So we're just drawing shapes at the moment, although this is supposed to represent the rough chemical shape of that sugar. And so we have sugar, phosphate, and base. This is our nucleotide. The bases in DNA can either be A, G, C, or T, adenine, guanine, cytosine, and thymine, and it can be U in the case of uracil. T is replaced with U in the case of RNA, like that. So these are the DNA. RNA uses U instead of T, but it still uses A, G, and C. The sugar, when we're in DNA, we call it deoxyribose. Whether or not you would need to know that for a question is pretty slim. It just means that ribosome, deoxyribose, this just means that it's missing an oxygen. Whereas with RNA, it's just ribose. So they're almost exactly the same sugar. And then this here is our phosphate group that we saw, exactly the same as the phosphate group that is in the plasma membrane as well. And so then what we can do is we can look at this in a little bit more detail. If we look at a phosphate group, it's got an oxygen hanging this way, an oxygen that way, an oxygen that way, double bonded oxygen this way. This then glues on. You would not need to know how to draw these, by the way, but CH2 like this. I think it's got an oxygen at the top. And then these, it depends on what kind of base you're looking at. They have slightly different shapes, like this. And then this would be our nucleotide in a little bit more chemical structure. If I were to copy that, let's just do that, and then have them facing each other. This is what a base pair in DNA actually looks like. One's pointing up, one's pointing down. And between these bases here, this might be an A and this might be a T, then we would get some hydrogen bonding between their compounds like that. Again, we don't really need to go into the chemistry of it in this one here. I already cover some of the hydrogen bonding and stuff like that in the crash course video. But this is what's going to create the ladder. So this is the rungs of the ladder here, and this is the struts of it, or the backbone on either side. More importantly is what is happening in the base pairing. Only A and T can go together, and only G and C can go together. I can never remember which one's which. I'm pretty sure I've got the A and the T the wrong way around. I think A actually has one ring and T has two. Can't quite remember. But these two go together, these two go together, and this is referred to as complementary base pairing. And this is half of the challenge in the GAMSAT questions, is just understanding that code. It can do a lot for you. So from there, if we were to zoom out a little bit, you can imagine that we could just have lots and lots of these base pairs going on, and all of a sudden, see how they fit together, and we're getting that ladder structure. And what's happening between the phosphates and the sugar is you're forming a bond between each of them, like this, and gluing it all together, and you get this double helix. And that is our DNA structure. What we're most focused on is what's happening in the middle here in terms of the base sequences, like that. Cool. So that's where we kind of head to now. So if we look at complementary base pairing a little bit more, so it's A to T, G to C, like this. And the idea is that if we have a sequence of DNA that goes A, G, C, T, T, A, G, C, C, A, well then the side that would be attached to it, the complementary side, would be the opposite. So A binds to T, G binds to C, C binds to G. And you can start to then translate it quite quickly. And that's something that you can just practice. The other little thing in terms of notation is you might see 5' and 3' written on there like this. And what that refers to is which end of each of the sides it actually is. An easy way to tell is the 5' end has the tail on it, the phosphate kind of hanging off of it. It's a larger number. And the 3' is the blunt end, like that. And likewise. And you'll notice that they're anti-parallel. They run opposite to each other. You won't have them pointing in the same direction, always opposite. And so therefore you can use that kind of symmetry to do a lot of problem solving as well. That's how I've known that if one of them is 5 to 3, the other one must be 3 to 5. And there we go. So that's our complementary base pairing. I'll point out as well, I was going to do it as a question, but I'll just do it as an explanation. Keep in mind that if they tell you the percentage of one of the bases, you can work out the percentage of the other three in a piece of DNA. If 20% is made up of A, there must be 20% of T since they bind together and they go in pairs. That then leaves behind 60% to be shared between G and C. Since they've got a base pair, it's got to go evenly, so therefore it's 30% G and 30% C. Always look out for that one. They used to have that in the old sitting, like when I sat it in like 2012. I remember getting a question like that. And it's a nice, easy one to grab if you know that trick. Cool. And I think that then is everything that I wanted to cover in this particular crash course. So what we can do now is go through some of the questions. Hopefully that wasn't too rapid for people. Okay, so what we're going to do is now go through, I've written up some sample GAMSAT questions that then apply this knowledge so you can get a sense of, even though we've gone through all the content and everything, it is a reasoning exam, but you need to use some of that scientific knowledge to be able to actually work with the information. So this one here that I've written, DNA probes are short strands of single-stranded DNA that contemporarily bind to complementary sequences in a sample of DNA for detection and identification of a particular base sequence. Probes often have a fluorescent protein or radioisotope attached in order to be detected. One particular DNA probe utilizes the fluorescent protein GFP found in some aquatic species. GFP, when exposed to blue light ranges or to ultraviolet ranges of light, a lot of typos in there as usual, will exhibit bright green fluorescence, 550 nanometer wavelength. The target sequence of the DNA probe containing GFP is blah, blah, blah. And then we've got fluorescence of the protein containing probe is usually achieved through photodetection of reflected light onto a photosensitive surface, which generates a graphical output. An example of such an output is shown in figure 1 below, where an abundance is measured in arbitrary units and is proportionate to the amount of DNA hybridization that occurs within the sample, blah, blah, blah. So I'm trying to kind of overwhelm people with the amount of knowledge, but really, we've only got a few things in here. So we've got, this is the sequence in the GFP probe, and it goes bright green, 550 nanometers, and they're using the label GFP. And then we get a graph. All of this is just a fancy way to say we get a graph like that. And this is an example of one of them. Question 1 then says, a sample of DNA is being tested for the presence of a particular base sequence using the GFP carrying DNA probe. Which of the following sequences would this probe be most appropriate at detecting and binding to? So if this is what it has, it needs to be complementary to that. So this is an example where we can solve the answer. So they're all in the 5 to 3, so I'm going to stick with that. That would mean they would have to be complementary to this, so I'll just switch colors here. That would have to be 5, A would be T, C, A, G, G, and then 3'. And I just look for that, T, C, A, G, G, and the answer is C. Simple as that. You can see having a knowledge of what this actually means that you can speed up your question answering. That's the thing that we want to get out of this. The next thing then is, which of the following would be the most accurate depiction of the output graph for a sample of DNA binding the GFP carrying probe? Now I'll zoom in a little bit here. You can see that they're effectively all got the same axes, and the wavelength is measured in by 10 to the negative 7 meters, which is 100 nanometers. You could think of it that way. So that means that 1 actually represents 100 nanometers, and we were told that the GFP shows a signal at about 550, so that would be 5.5 on the scale. So we can see here this one's got a peak at 3, so that's not it. This one's got a peak at around about 5-ish, so that's a possibility. This one's got a peak at around about 5.5, there we go, like that. So this is looking pretty good, and this one does not have a peak. It seems to be peaking way up at 800, which doesn't make any sense. So this would be your best option. The actual shape of the graph doesn't matter so much, because that would be about how much it also reflects. We were told that it reflects bright green light. It may reflect other colors either side of it, but the actual width of the graph is irrelevant. It's all about where that peak is occurring. Cool. Okay, so in question 3, we've got another probe carrying now the blue fluorescent protein. BFP exhibits a bright blue color at 380 nanometer light under UV. This one has a sequence of AAA CGT in the 5 to 3 direction. And then we're told if a sample of sequence blah, blah, blah is also treated with both of these probes, which of the following would then show their graphs? And ACER love these types of questions where you're comparing the graphs and they're changing one little finicky detail at a time, and you're looking at relative changes in them. So the first thing is, because the probe sequences are smaller, I would first work those out. So if this is the probe, I need my sequence to be complementary to it. So complementary to this would be 3'. This would be T, 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 and then G, C, A in the 5 to 3. And I'm going to go and see if I can find that anywhere. Okay, and so we can find that one there. So we're looking for T, 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 G, C, A. And I can see that just here, like that. And then for the green fluorescent protein, this one, its complementary strand was GGACT in the 3 to 5 direction, because that's the direction that we've actually got the sequence given. So GGACT, I'm going to look for that one. I can find GGACT just there. Oh, I'm supposed to switch colors. Green, GGACT. I'm going to keep scanning, looking for GG. There's one there, but it doesn't follow with GGACT. But there's another one, GGACT. And that's it. So what that means is that the green fluorescent protein will bind twice, whereas the blue will only bind once. And we're told in the question that it says in the stem, the amount of arbitrary units, or the abundance, is proportionate to the amount of DNA probe hybridization that occurs. So the amount that it binds to the sample. So what that would mean is that the height, the peak of the green should be twice the height of the blue. And then we also got to line up their relative wavelengths, although if we look at them, all their wavelengths are all lining up the same, so we don't really have to test that one. And instead I'm just going to look at where the peaks are, and I can see the solid line GFP should be twice the height. That looks pretty good. And this is lined up at 380 roughly, and this is lined up at about 5.5. This one over here, these are equal, so that's not it. This one here has got it the wrong way around. It's put the blue as being higher, that's wrong. And this one here has put them as equal, just lower down. Remember, it's arbitrary units, the height doesn't actually matter, it's the relative height. So that's also wrong. So option A would be the best option there. And then finally, question 4, if the same sample of DNA were being tested during a mitotic division, which of the following would be true? So this is a kind of test and check method here. Abundance of the GFP would double during metaphase, but would not change for BFP. So if we think about it, abundance was about the amount of detection, the amount of that sequence in there. During metaphase, you've got twice the amount of genetic information, same number of chromosomes, but twice the amount of genetic information. So if you're testing it, you should get twice as much detection. So you should get twice as much GFP. So this sounds pretty good, but then it says it wouldn't change for BFP. That information is still in there as well in the DNA, so that should also be doubling. So this is the bit that is wrong there. Abundance for both would double during mitosis. This is technically true, but it says during mitosis, and there are going to be phases in mitosis where the cells are actually separated in some way, right? And so yes, it does double, but it may not be double all the time. So it would be a maybe at this point. With C, abundance for both GFP and BFP would not change. Absolutely not, because then you've got twice the amount of DNA, so you should get twice the amount of detection. That bit is wrong. Abundance for both GFP and BFP would vary at different stages of the division. So in that case, that one would be kind of true, because depending on which stage in the mitotic division you're grabbing the cell at, it's going to depend on how much genetic information is there. If you've got it in the early stages, it's still only got just the initial amount that it had. If you've got it after replication, then you've technically caught it with twice the amount. If you've caught it after cytokinesis and telophase, where the cell is separated, it's got the same amount as it had initially as the parent cell again. So it depends on which phase you're catching it at, which is really just testing if you know when in the cell cycle the DNA is doubled versus when it's not. That's it. So that would be the most correct. If you were to overthink it, I do notice there's a slight weakness in the way that I've worded that. During mitosis is a little bit vague, but again, you always go on the one that has the most evidence going for it. Cool. All right, and there we go. So hopefully that was really helpful to kind of give a relatively rapid run through of everything that you need for the cell structure and cell cycle, including cell division and a little bit on DNA. This is hopefully giving you some tools to now work through some more bio questions. Again, bio is a bit of a weird one. A lot of the information you'll get thrown is going to be pretty new, but some of these key principles are things that carry throughout. Obviously, they come from first year bio, and yeah, it's really just those few things that I've highlighted in the questions that probably pinpoint the most important elements of the biology stuff that you need for this particular subtopic. In the next one, we're going to go through all of the systems of the body, which should be a pretty large video, I'd imagine. Maybe I can cut it down a little bit, but we're going to go through the cardiovascular system, circulatory system, respiratory, intestinal or digestive system, urinary system, reproductive system, all of those. Only in the context of GAMSAT though. It's not going to be a big, massive, content-heavy one. It's just going to be what are the key things that we can pull from them, what's their main function, the position, like the organs that are involved and how they interact with one another and that kind of thing. So that, so you can come away with some useful information, particularly the cardiac cycle, because that's the one that they love the most. Anyway, that is a long enough video from me, I would imagine. So I will see you guys in the next one.